So, good morning. My name is Danny Hoyer, from Melbourne, originally from Basel, a little bit of all over the place, University of Melbourne and the Floyd. So it's my great pleasure and privilege to introduce you to Professor Sarah Tabrizi from the UCL. So Sarah is the director of the UCL Huntington's Disease Center. She's also a joint head of Department of Neurodegenerative Diseases at UCL Queen Square Institute of Neurology, a principal investigator at the UK Dementia Research Institute, and a consultant neurologist at the National Hospital for Neurology and Neurosurgery. And in addition to a basic bench science program focusing on basic cellular mechanisms of neurodegeneration in Huntington's disease that can be harnessed for therapeutics, she's also leading a large translational research program that is working towards finding effective disease-modifying treatments for Huntington's disease, which is quite important. As you know, there's almost nothing there for the time being. She was global clinical PI on the world's first gene targeting study for Huntington's disease using antisense oligonucleotide therapy. And so it's my great pleasure to have Professor Tabrizi on the show.
Thank you, and thank you for inviting me to come and tell you about what we're doing in Huntington's disease. And I'm from Edinburgh, so it's very nice to be back in Scotland. These are my disclosures, and I'm on the scientific advisory board for quite a lot of industry partners who I work quite closely with, testing molecules in the lab. But everything goes to UCL Consultings, and I don't take anything personally. So I work on Huntington's disease, and this is a devastating genetic dementia. It's the world's most common genetic dementia, and this is a 38-year-old lady, and this is her 13 years later with end-stage disease. It affects people with a mean age of onset of 40, and it's fully penetrant, autosomal dominantly inherited, and incurable, and it's caused by a CAG repeater in the first exon of the Huntington gene, which encodes a protein mutant Huntington and causes disease through a predominant toxic gain of function. So this is a lady with Huntington's disease, and she's got widespread movements, which is chorea, which comes from the Greek word to dance. She has evidence of dementia. She's dystonic. And so people with Huntington's disease in adults are in these constant movements. And actually it affects young people. This is a young man, child I looked after. He developed the disease when he was 12. He died when he was 20. His mum, who's standing behind me, lost her husband when he was 33. And Emma's sister, Cherry, died when she was 27. So this woman lost her husband and 2 kids. So when you work with Huntington's disease families, which I have since my PhD, you become absolutely passionate about trying to develop a treatment. And so this is an overview of potential therapies that are in development. And this is a kind of overview of what is important in Huntington's disease, but also is relevant for all inherited repeat expansion diseases. So I'm going to talk about a little bit about a whole new area that has really exploded in DNA repair modulators, better known typically in cancer, but they're now big in neurodegeneration. I'm also going to tell you the story of the antisense oligonucleotide and tell you a little bit about some preclinical programs that are moving forward. So it's going to be in 2 parts. There's going to be the challenges, which is the ASO story, which has had its ups and downs. And I'm going to tell you about the opportunities, particularly in DNA repair, gene therapy, allele selective targeting, which is targeting only the mutant gene, and a little bit on other diseases and why this is important. So Huntington lowering. There are over 50 papers now published showing that if you lower mutant Huntington in a mouse or rodent model, either genetically or through siRNA or antisense, you can reverse symptoms of the disease. So the evidence for Huntington lowering is overwhelming at the preclinical level. It is not that difficult to cure a Huntington's mouse. But the problem with human brain diseases is this is a mouse brain, this is a rat brain, this is the largest rhesus monkey brain, and this is our brain. And you can see the challenge we have here is delivery, delivery, delivery. We don't have ways or means to deliver molecules yet uniformly to the whole human brain, particularly genetic therapies. And so antisense oligonucleotides, these are single strands of chemically modified DNA, and they've been modified to stabilize them, and they bind to their cognate mRNA through Watson and Crick base pairing. And it's a very specific binding. And that leads to a hybrid, which is then degraded through the RNase H1 mechanism. And that very simply results in reduced translation and less protein being made. And so those who are interested in structure, I'm going to tell you about MOE. ASOs, they have an MOE moiety at either end, which gives it affinity and stability and tolerability. And it has a mixed backbone of a phosphodiester and phosphorothioate linkage that allows it to be recognized through the RNase H1 mechanism. And it's a 20 base pair oligo. So basically, it's a piece of DNA that's been stabilized. And so I started working on this program in 2010. In close collaboration with Frank Bennett from Ionis. And this is some of the preclinical work. We delivered the ASO intrathecally because we wanted to develop an intrathecal protocol to deliver it, which is basically a lumbar puncture. We tested intracerebroventricular and we tested intrathecal. And the data, this is just a summary of some of the preclinical data. Intrathecal delivery RNA protein, we had very good lowering in the frontal occipital cortex. The deep structures, which is the caudate and the thalamus, about 50% lowering. And here is a single dose, and it lasts about 8 weeks. So it's got a long pharmacodynamic effect. In the caudate, maximal lowering of 50%, but then it begins to come up. And the caudate's a very deep brain structure. And so that's one of the challenges of delivery in the antisense oligonucleotides or any nucleic acid delivered intrathecally has a predominant cortical pharmacodynamic target engagement effect. And the toxicology program identified a really wide safety margin. It was squeaky clean. Histopathology was normal. Histology was normal, and there was a very wide safety margin for going to the clinic. So we developed an intrathecal protocol because actually an antisense delivery hadn't been done in adult humans before. So we developed an intrathecal protocol where we took off 20 mils of CSF and then delivered the drug in 20 mils of CSF over 3 minutes with a lumbar puncture with a fast push bolus. And that was modeled using non-human primates with pressure jackets to raise the heart rate because your CSF flow is dependent on heart rate. So this was developed to see if we could give an intrathecal injection and get the ASO to the brain in someone with a spinal cord the length of a human, which is about 50 centimeters. So we didn't know if this would actually work, but it did, in that the ASO got to the brain and lowered mutant Huntington in the CSF. And this was the result that everyone got quite excited about in 2017, which was dose-dependent lowering of mutant Huntington in CSF. And so this made headlines all over the world. Everyone got super excited. It was the first ASO trial that had been shown to lower the level of a toxic protein in adults with a neurodegenerative disease. And everyone in the HD community thought a treatment was around the corner, but not so. But Roche bought the program and decided that the phase 1 data was so promising that they would go straight to a phase 3 study because they felt that they had evidence of target engagement. And the late phase team wanted to go for maximum tolerated dose to try and get as much as possible to the striatum because delivery is often dependent on dose, and so the higher the dose of a nucleic acid, the more you can get to deep structures. That was the theoretical basis for the choice of 120 milligrams. 120 milligrams was the top dose. So a phase 3 study was started with 791 participants testing the ASO tominersen at 120 milligrams every 8 weeks, testing 120 milligrams every 16 weeks versus placebo in people with late symptomatic disease, which is a bit like the lady in the video I showed you. And that was because they wanted to do just a 2-year trial. And this was the fastest recruiting trial in the whole history of Roche. And Huntington's disease is a rare disease, but this was the fastest recruiting trial that they had ever had. And then in March 2021, the trial was stopped by the Data Safety Monitoring Committee. And it was stopped because the active group at the highest exposure were doing worse than the placebo, which was devastating for the HD community. And this is some of the data. This was one of the primary endpoints, the total functional capacity. This is the 8-weekly group. This is the 16-weekly group. And this is the placebo. And this is the placebo 16-week, 8-weekly group. This is when the trial was stopped at week 69. And you can see here, they're doing worse. Also, what was seen is the brain ventricles were expanding. Although there wasn't an associated increase in brain atrophy, because if your ventricles expand, typically it's associated with neurodegeneration or aging, and there's an increase in brain atrophy. This was expanding ventricles, but no increase in brain atrophy, which suggests a kind of dynamic effect. And you can see here, placebo, 16-week, 8-weekly group, there was a 24% increase in brain ventricle size in the 8-weekly group, 17% in the 16-week, and 11% in the placebo. And that's, the placebo is what one would expect with neurodegeneration. But this is a large increase in ventricles. And this is, it's a lot of data on this graph, but I'm just going to talk you through it. This was the original phase 1. And many of us who were closely involved in this study were not keen on the 120 milligram dose because if you can see here, CSF neurofilament was going up in that group. So a number of us were a little worried about the CSF neurofilament going up because it suggests CSF neurofilament is an neuroaxonal marker typically associated with neurodegeneration, never before recognized as a safety biomarker, but if CSF neurofilament goes down, it's thought that it's rescuing neurons, and it goes up normally in neurodegeneration. But this was already something that made us concerned about the 120 milligram dose. This was the open label extension. The subjects had loading. So they had 2 months of loading of 240 milligrams in the phase 3 as well. And you can see here, there's a spike up in CSF neurofilament in the monthly and in the 8-weekly. This is the Generation HD1 data. This is the CSF NFL. This is protein, ventricles, and mutant Huntington. This is placebo, and you expect some increase in the ventricles in placebo and with the NFL. But you can see here in the highest exposure, huge increase in CSF neurofilament, big increase in ventricles, an increase in CSF protein, and dramatic lowering of CSF mutant Huntington. So this is going from low to high exposure. Tau and YKL40. YKL40 is an inflammatory marker, and you can see here there's evidence of inflammation, and tau goes very similarly, and CSF protein goes way up, and CSF protein, you don't want high protein in your CSF. So, what went wrong? The dose. The dose was too high. And the dose was chosen by the late phase team with loading because they wanted to get as much as possible to the striatum, which is an important part of the brain targeted in Huntington's disease. And that's where I mentioned about delivery and distribution. I think this has been an enormous learning for the field. Also now, CSF neurofilament going up is now recognized by the regulators and everyone as a safety biomarker. Before, no one really knew what was CSF neurofilament going up. But now, if you do a trial and CSF neurofilament goes up, I would stop your trial. And actually, Novartis did that recently with branaplam. They stopped the trial. They halted the trial because CSF neurofilament was going up. Chemical inflammatory effects, that's what, with the high CSF protein and white cells, and you get impaired resorption of CSF, and so the ventricles get bigger. And this approach lowers wild-type and mutant, and it might be that there was too much wild-type lowering because it's a very effective ASO, but you can't really dissect that from this data. The lack of efficacy is complicated because there was evidence of toxicity. The patient stage was pretty late in disease, like the lady I showed you, when there's already widespread inflammation, widespread neurodegeneration. And the dose chosen, I think the effects are, it was the dose chosen was too high, too late stage disease. And I think this has been a big learning, and actually we organized a Royal Society symposium on oligonucleotides based a lot on this big trial stopping, where there was a lot of discussion from toxicologists, preclinical, about how do we do better nucleic acid therapeutic trials for the brain. And none of this was predicted from the 15-month, it wasn't just 3 or 9 months preclinical GLP tox, it was 15 months preclinical GLP tox safety. But one of the things in all of these is these are wild-type monkeys, and then you're injecting into a normal brain. And then you give a high dose of an inflammatory antisense oligonucleotide in someone who's had 25 years of neurodegeneration with inflammation. And I think we have to think about what are our models for toxicology. And there's a big interest now in organoids and brains on a chip to try and test some of this. One of the things that came out of this is that Roche did a hypothesis-driven post-hoc analysis and wanted to look if there was any younger patients who were earlier in disease stage who managed to tolerate the dose of ASO. And this is post-hoc analysis, so it could be completely meaningless. It needs to be replicated. But those who were younger, below the age of 50, with earlier stage disease, appeared to go in the right direction on all of the clinical measures. And this is those in green, the lower exposure. The other important finding is in the neurofilament. In the 16-week group, which is the lower exposure, the neurofilament in plasma is beginning to go significantly below baseline. That's suggestive that you're saving neurons. It's been shown in multiple sclerosis and ALS and SMA to be associated with clinical benefit. This is early. We don't know what this means. But I think importantly, we now know what caused that big neurofilament spike, because there were some subjects who were rolled over from a natural history study to get the drug at 16 weeks at 120 milligrams, but they didn't get the 240 milligram loading. And they had no NFL spike. So that 240 milligram loading, which was chosen by the pharmacology modeling team, was to try and get steady state as quickly as possible and then load and then give the ASO. And you can see it was the loading of 240 milligrams which caused the NFL spike. So if you didn't do the loading, there was no spike. So there is now a phase 2 dose-finding study. So I think there's a lot of learning here. There should have been a dose-finding study going from the phase 1 before the phase 3. But this is a lot of hindsight, and actually this paper is in press at New England Journal, and it's a learning paper for the whole field of nucleic acid therapeutics, I think, in terms of what not to do. Don't load, don't go for maximum tolerated dose, and you need to treat early in the disease. So now, and you need to do a dose finding. So now is a phase 2 dose-finding study, which is screening 100 milligrams every 16 weeks, 60 milligrams, half the dose every 16 weeks, and placebo for 16 months. And this is going to test in much earlier stage patients. So it's watch this space. And tominersen's a very good drug. It's just got a narrow therapeutic index. We're also developing approaches to target the mutant allele only. And this is very challenging because the difference between the mutant allele and the wild type is just the length of the CAG. Otherwise, they're identical. But we can identify SNPs that are linked to the mutant allele and design oligos against those SNPs. Now, they're more challenging because you don't have so much mRNA real estate to design your oligo, but it targets the mutant allele only. It is personalized medicine because it only can treat 40% of the population who carry that SNP. But these are moving forward for clinical development, targeting the mutant allele only and those who carry the SNP. So what does this mean? I think it means that we do need the next generation of RNA drug delivery, and this is a huge interest in the field with industry and within academia. We need better chemistry. It's all about delivery. We need better chemistry, better backbone, less inflammatory, possibly stereochemistry. We need conjugates, for example, antibodies, and possibly also lipid nanoparticles or antibodies or lipid conjugates. And this is a huge area of research that really it's a big area that many people are working on, conjugated siRNAs and also antisense oligonucleotides. And this is because of this. The CNS is this huge unmet need because targeting the human brain is a massive challenge because you need to target the whole brain to treat these diseases. So it's all about delivery and distribution. And so the MRC actually gave a consortium of us recently £8 million over 3 years to try and develop and look at delivery. And this is a number of colleagues. There's some industry colleagues. We're developing new linker chemistries. We're looking at uptake of nucleic acids, endosomal trafficking and escape in the big areas of unmet needs, CNS, heart, and skeletal muscle. And so this is a exciting collaboration, and I'm hoping that we'll come out with some better designed oligos. So now I'm just going to move to DNA repair. So those of you who work in cancer, this has been a huge topic in cancer for a long time, but DNA repair is now the hot topic in repeat expansion diseases. And so repeat expansion diseases as a whole are actually quite common. Huntington's is one, Fragile X, Friedreich's, myotonic dystrophy. There's 15 spinocerebellar ataxias that are repeat expansion. There are over 50 neurological diseases caused by repeats of DNA. And what we know in these diseases is that the repeats of DNA expand throughout lifetime. And this is a Huntington CAG repeat. This is 42 repeats. And this is age of onset. And you can see for 42 repeats, you can have an age of onset in your 20s right up to your 70s. And we know that the repeat, and this is another repeat expansion diseases, expands throughout life. So you're born with, say, 42, but in your brain, the CAG repeat is expanding in postmitotic neurons. And this is an old paper from Peggy Shelbourne, actually when she was based in Glasgow in the Department of Genetics, where she looked at post-mortem and found CAGs of up to 700 in the striatum. And myself and Darren Monkton in Glasgow have done some work showing that patients with more repeat expansion have faster disease, and people with less repeat expansion have slower disease. So repeat expansion looks like it drives the rate of disease. And then in 2017, one of my PhD students was doing a GWAS on a large cohort of HD subjects that we were following, and this came up, MSH3, which we didn't expect. So this was the biggest hit. MSH3 is a mismatch repair protein. It's key mismatch repair protein, and this was a powerful modifier of HD progression. Those who had the variant had slower progression. We also found that those who had the variant had reduced expression of MSH3, and reduced expression was associated with slower progression, and we found the same in the muscle disease myotonic dystrophy. And so what we think happens is MSH3 is part of a complex called mutase beta, which binds to these loopouts that occur in DNA, repeat DNA, and then another DNA repair part complex, MLH1 and PMS2, nick the DNA, XO1 cuts it out, and then POLG fills the gap, and the DNA and the repeat gets longer. That's how somatic expansion occurs, and it looks like it happens during Huntington transcription. We've done a lot of work knocking out the promoter. So if you target somatic expansion, it looks like you'll target one of the very root causes of the disease. You stop that repeat getting bigger and also targeting the protein itself. And we have been testing an MSH3 ASO. This is now moving forward for clinical development. And if you treat with an MSH3 ASO, you completely ablate somatic expansion in HD neurons. So it just stops it. If you knock out MSH3, you stop somatic expansion. So there's a huge amount of work now looking for molecules that target MSH3. Many people have programs with small molecules, because if you can have an MSH3 targeting ASO or siRNA or small molecule, you'll not just treat Huntington's somatic expansion, you can treat all the repeat expansion diseases. So it's a really big area. So I'm just going to tell you a little bit about a zinc finger transcriptional repressor that we're working on taking to the clinic. ASOs I've talked about, small molecules targeting DNA repair. We're also doing a gene therapy, which is being delivered to the striatum. Gene therapy also has the challenges. ASOs and nucleic acids get to the cortex predominantly. Gene therapy targets a focal region. You can't transduce the whole brain with AAV9. At the moment, it's targeting the striatum. And we are dosing a microRNA gene therapy into the striatum to switch off Huntington. And we dosed the first patient last year. We've now dosed four patients. It is a 17-hour surgery. It's really long neurosurgery. So each of these approaches may be used in tandem, but the gene therapy approach, we need better AAV9, we need better delivery to the brain, and ideally we need intravenous. And so I'd like to finish on telling you a little bit about this transcriptional repressor, zinc finger protein, that I'm excited about, that's an allele selective approach. Because wild-type Huntington's important, so the ideal treatment is just targeting the mutant and preserving the important wild-type. But as I said, it's challenging because they're exactly the same other than the length of the CAG. So zinc finger proteins have been engineered that repress the mutant Huntington only. It targets all the toxic species because it targets the DNA and all the splice variants. And it binds, and it preferentially binds longer CAG repeats through cooperative binding, and you get recruitment of multiple zinc finger protein transcriptional repressor fusion proteins. And this rep functional domain is actually the CRAB domain, which is a transcriptional inhibitor. So you bind expanded repeats and switch off transcription. So it targets the DNA and stops transcription and stops somatic expansion. It stops all the downstream toxic species. So this is, I'm more optimistic about this than using CRISPR, because CRISPR has this enormous challenging of getting the Cas9 machinery to the brain. So we know that the ZFPs can lower mutant Huntington in mice. It can reverse the PD10 PET loss in the striatum, and you reduce the Huntington inclusions. Again, the challenge is gene therapy. This is, again, needs to be delivered by gene therapy, and at the moment can just target the striatum. So ways of delivering this are still in preclinical development. The ideal thing about gene therapy is it's a single shot. This approach targets DNA and it's allele-selective. And at the moment, we're working on optimizing the cargo to mitigate off-target, promoters, and improved viral capsids. And as I said, the surgery at the moment is taking 17 hours. It's too long. And people end up with all the side effects of having a 17-hour general anesthetic. So this is a huge area. I think this is relevant for all brain diseases and is ongoing. So just to say, will these therapies work? We know they work in that this is nusinersen, this is Spinraza. Now, this is a very different mechanism of action. This is, Spinraza is replacing a missing good gene. So gene replacement is a lot easier than toxic gain of function lowering. And so Spinraza was designed to incorporate exon 7 into a functioning SMN2 protein, which is missing in spinal muscular atrophy. And this has been, upregulates the level of the good missing SMN2 protein. That has had miraculous effects for spinal muscular atrophy, which is a fatal infantile disease. And now there's Zolgensma, which is a gene therapy. This is an antisense oligonucleotide that has just been given accelerated approval from the FDA, tofersen. And this study missed its primary clinical endpoint, but the subjects all rolled over into an open label extension. And this is the concentration of NFL in plasma. This is the SOD1. This is the, and you can see here, NFL goes down when you go on the drug. And then here are the clinical measures. All the people on the drug in the open label extension were going in the right direction. But this has been a very important ruling by the FDA. They gave an accelerated approval for tofersen for SOD1 ALS based on this massive drop in NFL. And so even though the clinical improvement, it's a very rapidly progressive disease, ALS, did not show significance, the NFL was really striking, and they gave accelerated approval based on the biomarker. So I think for neurodegenerative diseases, this means a lot, because it means that if we can have a therapeutic that has benefit by lowering NFL significantly and has some evidence of clinical benefit, it may be that we get an accelerated approval. So this was a very important ruling. And then more recently, there's been a tau trial with ASO in mild Alzheimer's disease. And the reason I'm showing you this just got published in Nature Medicine. The reason I'm showing you this study, this is the Biogen tau ASO, is this. This is tau PET. This is screening. This is week 25 after being on placebo. In week 25, this is a male person. This is another 67, 71-year-old male. Then they went on to active drug, 115 milligrams every 12 weeks. This is week 100, and you can see the clearance of tau in the regions affected in Alzheimer's disease. So this shows you that the ASO is clearing tau in Alzheimer's patients. So I think intrathecally delivered ASOs have huge potential. They're in trials now for Parkinson's. They're in trials for Alzheimer's. They're in trials for multiple system atrophy. There are numerous trials with these approaches and with conjugated siRNAs. And I think for me in Huntington's disease, I want to be able to treat people decades before they show signs of the disease. And we're doing a study. We're studying people who are 20 years old, who are 25 years before they're predicted to show the disease. They carry the gene. And that's when we want to treat. Because if we can treat then, because there's absolutely nothing to find at that stage of disease, their brains are completely normal. And if we can treat then, my hope is that we will prevent Huntington's disease ever occurring. And that's actually a big Wellcome Trust program that is moving forward to try and do prevention treatment in very young HD gene carriers, which is our hope for the future. So I'd like to thank, this is all of my research group, and I'd like to thank you for listening.
Thank you, Sarah. This is a fantastic presentation. Now, history repeats itself. It seems that industry just never learns.
They don't.
Same thing
They
happened.
are listening now, but they didn't.
Same thing happened in Alzheimer's disease, where they were treating too much, and you have 100% or 90% reduction in amyloid, when actually what is wrong with amyloid is only about 30%. And if you were to stick to this one, maybe you have a chance.
Oh, you're absolutely right.
Now, the other thing is, we had a Fragile X program where, you know the number of repeats, and then this patient is going to have the full-blown thing by that age, and etc., like in Huntington's. Now, here comes the surprise. We ended up with a pair of twins. One was absolutely normal by all standards. The other one had full-blown disease with the same apparent number of repeats. How do you explain that? But anyway, I see that our president has questions, so please.
Can you hear me?
Yes.
Yes, okay. So that was an absolutely fantastic talk. Great update for somebody outside of the field. So I notice you're talking to Alnylam and you're working with Silence and UMass and Ionis. So presumably you're interested in small interfering RNA.
I am. Yeah.
And clearly the problem there, as you've said, is that targeting is going to be awfully difficult. But is there a particular advantage of going with an siRNA approach, do you think, to knock down?
It's a good question. So I think the advantage, the advantage is that engaging the RISC complex is easier than RNase H1, and you tend to be able to go with much lower doses, and they tend to be more potent. There is still the risk of inflammatory effects with all these molecules, but siRNAs are easier because I think that because you engage the RISC and they're more potent. So you can go with much lower doses to try and get deep brain penetration. So Alnylam, we're about to start a first-in-human study with Alnylam with a C16 conjugated, which is a lipid conjugated siRNA against Huntington, which is very potent. So I think the challenge there, because all these approaches are non-allele selective, is that we don't lower wild-type Huntington too much, and we obviously monitor for inflammatory effects. So I think there is, and we're also doing, Craig Mello and Atalanta have developed divalent siRNAs, which are two siRNAs conjugated together. So we're going forward with those approaches as well. So you're absolutely right, David. They are tend to be more potent, and that you can use much lower doses, but they still have the risks. It's a bit like the story with patisiran. So Alnylam have this developed GalNAc to siRNAs to target the liver, which have been a revolution, and treat TTR. It's actually been a cure for TTR. But when they did their first study pre-GalNAc, it's quite a long time ago now, they did a phase 3 with the pre-GalNAc siRNAs, and in the phase 3, 2 patients died. And so they went away, came back 2.5 years later with GalNAc conjugated siRNAs, which have been a revolution in getting delivery to the liver, and then developed patisiran, which has been huge for transthyretin amyloidosis. So it's a kind of lesson about the conjugate. And so that's, I think, the lesson. It's taken a long time to develop the C16 siRNA, but I'm optimistic about that one because we can go with very low doses. And also the learning from generation HD1 now is that everyone knows that you shouldn't load for the reasons loading with nucleic acids is not the right thing to do, and you need to be very careful about dose. So the thing about Generation HD1 is that it has resulted in quite a lot of learning for the community, and also, as I mentioned about Novartis, Novartis halted branaplam after 3 months, the trial, because the neurofilament was going up, and they halted it based on that, which I think, these things are all additive in learning about this. But yes, you're a very long answer, but yes, they're more potent.
Okay, next question.
Jules Heuberger from Center for Human Drug Research in Leiden, the Netherlands. Thank you for your very interesting talk on this very puzzling case, I would say, of the tominersen trial. Together with my institute, I was involved in the PKPD part of the study that Roche added later on.
Oh, yes,
but in the very early stage, the study was halted because of these findings. And you say that they should have done a dose-finding study before going into this, and I'm all for a dose-finding study, but I wonder how would that have helped this trial, because I think your story to me shows that we actually don't really understand sufficiently yet the disease and how the drug is influencing the diseased brain. Because you show that actually the exposure response to mutated Huntington is very clear. So how would a dose-finding study then come to a different dose than what they have done actually in the trial at that time?
It's a very, very good question. So in 2017, when we were trying to design what to do next, when it was still with Ionis, we had decided, and Frank Bennett, who's the chief scientist from Ionis, is very, very smart, knows a lot about ASOs, developed them. And we decided to do 60 milligrams every month. That was the dose we came up with, 60 milligrams every month, or 90 milligrams every 2 months. And this was based on what we had in the phase 1, which are much, much lower than what Roche chose, with no loading. And we wanted to do the trial in very early stage patients, so prodromal patients, i.e. hardly any motor symptoms. And that all got pushed out when the program moved to late phase Roche, because they weren't familiar with HD, they wanted a 2-year trial, they wanted a big trial, and they wanted to go for symptomatic to get a wide label. It's a long story, but it's kind of what surgery was saying about big pharma sometimes. I think if we had gone with the 60 milligrams or the every month, or 100 milligrams, or 90 milligrams every 2 months, and I think if we'd gone into prodromal, because if that post-hoc analysis is right in any way, then it means that lower exposure and much earlier stage disease, you may have a benefit. So I think what the 2 things that ideally should have done was to test much lower doses to try and understand the therapeutic index, and ideally have gone in much earlier patients. And I think what often happens is people are in a hurry, or big pharma, or they're in a hurry to try and get a result, or try and get accelerated approval, or try and do this. And actually, these neurodegenerative diseases are very complex. And I think one of the things that Generation HD1 has told us is you cannot treat late in the disease. I actually think by the time someone shows symptoms of Huntington's disease, it's too late to treat. I think we have to treat well before, when the brain has much more resilience and ability to recover. So I think it isn't just a dose finding, you're right, it's actually knowing the best time to treat. And I think that's the 2 things. But I think the reason the prodromal study didn't go ahead was because to do a prodromal trial would have meant larger numbers of patients over a longer period, and that's not what they wanted to do.
And how do these 2 things relate to the animal studies? Were these animals that you say were actually cured, let's say?
Mice. It's a good question.
Were they already showing symptoms, 1? And 2, how much of a decrease in mutated Huntington did you see there? Because still, to me, I mean, the fact that you lower with a higher dose, you lower more of the mutated Huntington.
Shows that mutant Huntington that's exactly right. The very high dose had spectacular mutant Huntington lowering and the patients got worse.
Yeah.
So there is it's never going to be a clinical surrogate. But in mice, and this is important, if you treat, and I think the best experiments have been done with genetic knockdown, if you treat a mouse at 4 weeks, 3 months, 6 months, 9 months, 12 months, 18 months, or 24 months, an HD mouse, at 4 weeks, 3 months, 6 months, or 9 months, you get really good reversal of neuropathology. If you treat at 12, 18, or beyond, you hardly rescue at all. So the preclinical models have already told us you need to treat much earlier. But one of the challenges here is the regulators wouldn't let us all do trials. When we did the first ASO trial, they wanted us to do it, this was in 2013 when we were talking to the regulators, they wanted us to do it in advanced patients because they said, well, we don't know if this is going to be safe. This was, there was a 24-page patient information sheet, including the risk of death. And so we ended up going for early-stage patients, but it was a big fight with the regulators and the ethics board, and also to include a placebo group, because they said, no, it's got to be late-stage patients to test this. The regulators now agree we need to treat early, but it took a long time to get them on board to say, because they didn't want to treat people who were otherwise well with what they thought were high-risk therapies. So it's actually now been a kind of building up of evidence. And now, actually, with the new HD staging system, we can treat before typical motor onset, and now trials are happening in that group. But in 2017, if we tried to design a trial in pre-symptomatic patients, we wouldn't have been allowed to. So I think every bit of, it's been a long journey, but the thing about the dose finding is that I think you would have had a better idea on risk-benefit and loading and the very high dose.
All right, thank you.
Yeah, good questions, thank you.
Well, keep in mind that industry has been obsessed by efficacy because many, many compounds have failed, classical approaches. And so you try to go to the highest dose that is tolerated. And this is certainly not to do with this kind of approaches, because if you knock out pretty much everything, including the normal ones, then you're in trouble. That's happened in Alzheimer's disease, it's happened here, and it's going to happen again and again.
That's exactly right.
It's learning on the fly.
It is.
Anyway.
They wanted to go for maximum tolerated dose. That's what I was told after.
Yeah. Well,
because they said that's what we do.
Yeah, and even with low molecular weight compounds.
And you can't do that with nucleic acid.
It doesn't work.
No, you're absolutely right, Sergey.
Anyway, I think no more questions. So we thank you again. It was fantastic.
Thank you. You're right. Yeah.
Yeah.